Dear learners, it is a part 2. In this part, we will discuss in detail the general account of the bacteria and their economic importance. I want to discuss the eukaryotic and prokaryotic bacteria, eukaryotic cell and prokaryotic cells. The eukaryotic cells are prokaryotic cell are smaller and have to simpler structure than eukaryotes. A nucleus and other membrane bound organelles are found in eukaryotes but not in the prokaryotes, and rod shaped chromosomes are found in the eukaryote. So, it is a general feature of the eukaryotic cells. Basically, they are the membrane bound organelles and rod shaped chromosomes which are found in the eukaryote. But in case of prokaryote, it would be different. The bacteria will come in the prokaryotes. So, in case of the prokaryotes, and their DNA are not enclosed by the membranes. Another thing is very important which are found in the eukaryotic histone protein. So, histone protein are not found in their the DNA. Histone protein they are able to make more complex of the eukaryotic cell. It is not found in the prokaryotic cell. And cell wall contain the complex. The very complex cell wall are there of the eukaryote. They contain the polysaccharide and such as peptidoglycans. In cyanobacteria and prokaryotes are prokaryotes. These are able to synthesize the food with the help of the photosynthesis. It is the comparison of the bacteria, prokaryote, archaeal cell and eukaryotic cells. So these are the number of the things we can compare it the different type of the cell like eukaryotic cell, prokaryotic cell and prokaryotic archaeal cells. So you can see in this table, it is the structure. Various structures are there like cytoplasmic membrane, nucleus, DNA arrangement as a true chromosomes and ribosome, cell wall. So these are the various things related to the structure. It is a prokaryotes. In case of the prokaryotes, the cytoplasmic membrane which are available in case of the three cells: prokaryote, archaeal cell and eukaryotic cells. And in some cases these are the not available nucleus, nucleus containing the nuclear membrane and surrounding DNA which are present in the eukaryotic cell but not in prokaryotic cell. So these are the various differences in the three type of the cells are there. Ribosomes, 70s ribosome which are present in eukaryote, but in case of prokaryote the 80s ribosomes are there. Or here is the 70s ribosome which are present in the prokaryotic archaeal cells. Cell wall constitute the peptidoglycan, pseudo peptidoglycans which are present in the archaeal cell, prokaryotic archaeal cell. It is present then the cellulosic. So these are the various things are there. You can compare with your three type of the cells are there. The general another thing I want to discuss, the general characteristic of the bacteria. So some characteristic like shape and size of the microorganisms are there, or bacteria are there. So I want to discuss one by one this size. The bacteria from vary from 0.2 micrometer to 2 micrometer. In cocci, it is another form of the microorganism, are between 0.5 to 3 micrometer. Bacilli range from 0.2 to 2 micrometer diameter and 0.5 to 20 micrometer in length. And Vibrio range, Vibrio is another type of the microorganism are there. Their range from 0.2 to 2 micrometer diameter. And Spirula In their size it is very big, 0.5 to 100 micrometer in length. Spirochetes in another, it is another type of the microorganism, range from 0.1 to 3 micrometer in diameter and 0.5 to 250 micrometer in length. So you can see the diameter and length of the microorganism. You can distinguish it by utilizing of the various shape and size of the microorganisms. By naked eyes we can see object is more than 200 micrometer. It is the shape of the microorganism. You can see it, the various shape are there. Coccus shape, you can see this figure, coccus shape, it is a round, you can see it, it is a round shape. Cocci, a plural form, and coccus is berry or spherical. You can see the structure of the coccus. Another is bacillus. Bacillus meaning rod shape of the microorganism are there. Relatively straight rod shape microorganism. Vibrios, you can see it, it is a comma shaped. Curve or comma shape of bacteria are there. Basically, Vibrio cholerae they can cause the disease like diarrhea. So, you can see the structure of the Vibrio. And spirulum are rigid spiral or helical form. So you can see it, the structure of spirulum, the bacteria, it would be the spirulum shape. Spirochetes, it is a spirochetes. The spirochetes are flexuous spiral form are there. So you can see it, spirochete, spirulum, vibrio, it is a fusiform between both the rod shape and coccus. So it is another very important form of microorganism that is known as the fusiform. So it is the shape of the bacteria are there. Another is arrangement of the bacterial cell. So how they arrange to each other. The pathogenic bacteria, sphere, cocci, rod, bacilli, and spiral. The cocci arrangement. So you can see it with the help of this figure. It is a coccus, it is the streptococcus and staphylococcus, diplococcus, tetrad, and sarcina. So these are the different arrangement of the microorganism. Cocci shape, diplococci. Cocci may be arranged into the in a pair, diplococci, and long chain streptococcus and enterococcus. It would be in long chain, division in one plane, and grapes like cluster. Staphylococci, when cocci divided into the random plane, and tetrad. You can see the structure of the tetrad. Here is the four cocci, they are arranged to each other, that is known as the tetrad. A square group in four cells when the cocci divided into two plane, genus Micrococcus. And cubical packet, you can see it is it contain the 8 cell in genus Sarcina when the cocci divide into the 3 plane. So these are the different plane, and they are able to divide it into the different here is the one plane, here is the random plane, and it is in the two planes are there, here it is the three planes are there. So ultimately you can see the different arrangement of the cocci are there. Bacilli arrangement, it may be flat, rounded, chain, and cigar shaped and bifurcated. So it is a arrangement of the microorganisms are there. Another thing I want to discuss, the cell wall of the bacteria and its adherent, means different number of the adherent which are available on the cell surface. They can cause the pathogenicity of the microorganisms, and their function is very diverse. The cell wall of the bacteria is a complex semi-rigid structure that surrounding the underlying plasma membranes. It prevents cell from rupturing, maintaining the shape, and serve as a point of anchoring of the flagella. The flagella, they are anchored with the cell walls. And another thing, the component of cell wall decided the pathogenicity and protect cell from the toxic substances and site of action of the antibiotic. So there are the very important things which are present on the cell wall. it is a site of action of the antibiotics. It protect from the toxic substances, and it can cause the pathogenicity of the microorganisms. So ultimately, various molecules which are present on the surface of the microorganism, they can cause the pathogenicity. Uh, I want to discuss another type of the bacteria, not bacteria but other type of microorganisms are there, like Mycoplasma. It is a bacterial genus and naturally lack the cell wall. So in case of the Mycoplasma, they don't have the cell walls are there. The plasma membrane of Mycoplasma have lipid called sterol and protect from the osmotic lysis. So in case of the mycoplasma, their plasma membrane, they have the a specific type of the lipid that is known as the sterol. They protect from the osmotic lysis. Archaea bacteria have the pseudomurin N-acetyl elosa N-acetyl elosa minimeric acid. It but not it is a different type of the chemicals molecules which are present on the archaebacteria, but not it is not a peptidoglycan like the true bacteria. And two type of the lipids are found in the found on the surface of archaebacteria, archaebacterial plasma membrane. One is the glycerol dihydrogenol diether and diglycerol diphenyl tetraether. So these are the very important lipids which are found on the archaebacteria. Another thing, there are the two very important type of the microorganism are there. One is the gram positive bacteria, second is the gram negative bacteria. So you can see this figure, it is the gram positive bacteria are there. You can see their figure, and it is a gram negative bacteria. The gram positive cell wall consists of a single thick layer of 20 to 80 nanometers. So their layer it would be very thick, it around 20 to 80 nanometer of the peptidoglycan, which are present outside of the cell membrane. The gram positive cell wall are stronger than the gram negative ones, and because the peptidoglycan layer, and gram positive bacteria um, sustain purple color, and gram negative bacteria stain pink or red red color by the gram stain. It is discovered in 1884. In gram negative cell, the peptidoglycan layer is the thin, about 2 to 7 nanometer, and surrounded by 7 to 8 nanometer thick outer membrane. So you can see it here is the another difference are there. You can see the peptidoglycan layer, it is very thick. Here is the very thin layer of the peptidoglycans are there. You can see it here is the wall teichoic acid are there. Wall teichoic, it is available in the cell wall. And another is lipoteichoic acid, it is connected with the lipids. So that is known as the lipoteichoic acid, which are available on the gram positive bacteria. So on the basis of this, the thickness of this, it is classified into gram positive and gram negative bacteria. Another thing, number of the other things which are present here is the lipopolysaccharides which are present on the gram negative bacteria. You can see this structure. So in this structure, here is the lipid A and core polysaccharide and O polysaccharide which are present on the surface of the gram negative bacteria. They can cause the pathogenicity. Ultimately, they are able to produce the antibody inside to the microorganisms, or they can cause the toxicity of the microorganism with the help of this O polysaccharide, core polysaccharide, and lipid polysaccharide. It is a cell wall, outer cell membrane, and here is the peptidoglycan layer are there. Plasma membrane, here is periplasm, number of the compounds which are synthesized in the periplasm, and phospholipid, it is the lipoprotein which are available in the cell wall. So these are the gram positive and gram negative bacteria, and their differences are there. Another thing I want to discuss, the surface adherent, the various surface adherent which are available on the surface of the microorganism. Many bacterial species secrete polysaccharide and small protein known as the glycocalyx, are small proteins. It is a viscous gelatinous polymer consisting of the polysaccharide, polypeptide found on the cell wall protect from the phagocytosis. So their role is a very important. They can protect from the phagocytosis and the surface adherent. They get not phagocytized by the certain cell, particular cells are there. And another thing, the capsulated, you can understand by the capsule. Glycocalyx is a described as a slime layer. It is a substance loosely attached to the cell wall. The glycocalyx of the vibrio permit to the cell to attach to the intestinal wall. So their role is very important, and you can understand the rough surface and smooth surface in case of the streptococcus are there. So you can understand the some microorganism are they cause the pathogenicity, and other microorganism they are the rough type are there. It is not pathogenic type of the microorganism. So it is totally depends on the surface adherent, glycocalyx and slime layer which are available on the surface of the microorganisms, or ultimately they are able to cause the disease. Your immune cell, they will not phagocytize. And another thing I want to discuss, the pili and six pillars. It is a usually longer than femori and 1 to 10 per cells. Pili transfer DNA from one cell to another. The role of sex pillars is very important. They can transfer their genetic material from one cell to other cell. Ultimately, the bacteria they get resistant, and some bacterial virus bind to the receptor on the sex pillars and begin to reproductive cells. So their role is very important, and the pili role is a very important which are available on the microorganisms. Another thing I want to discuss related to the flagella. Uh, Flagella, it is a filaments or to the hollow rigid cylinder, and constructed a single protein called flagellin. Their molecular weight around 30,000 to 60,000. So it is a very important proteins are there. Your flagella is made up with the flagellin. You can see this structure. So it is a bacteria, bacterial cell are there. It is a bacterial cell are there. It is a flagella. So you can, it is extended structure of the flagella are there. They have the outer membrane, peptidoglycan layer, and periplasmic space. You can see the periplasmic space of the bacteria here to the cytoplasmic membrane. So your flagella, they are anchored in the cell wall. So it is a various ring, P ring, L ring, and other rings are there, or ultimately flagella they will provide the movement to the bacteria, and some bacteria have sheath surrounding their flagella. Example, vibrio has membranous structure surrounding the filaments. Vibrio cholerae has a lipopolysaccharide sheath. The various structures are there which are available on the surface of the bacteria. Uh, uh, it is a team transmission electron microscopic studies of the bacterial flagella. It contains the three parts. First one is the filaments. You can see the filaments. Another is the hook. It is a hook part are there. Hook part. Uh, and third one is the basal body. So it is a basal body anchored to the flagella to the cell wall and plasma membrane. It consists a small central rod inserted into it, and it contains the series of the ring. So it is a series of the ring. It is inserted. It is a rod shape. It is inserted into the cell wall of the microorganism, or ultimately they will provide to the movement of the bacteria. Basically, it is a different type of the flagella are there. um, in case of the gram positive bacteria, the body has four ring connected to the central rod. The outer L and P ring associated with to the lipopolysaccharide. You can see it here is the L and P ring are there, which are associated with to the polysaccharide and peptidoglycan layer, while inner SM ring connected with to the PM plasma membrane. And gram in case of the gram positive has only two basal body ring, one an inner ring connected to the plasma membrane, and second an outer ring probably attached to the peptidoglycan. The flagella synthesize a complex process. 20 to 30 gene involved in the synthesis of the flagella. So one thing is very important in this case, flagellin protein, they are able to synthesize the flagella. The flagellin subunit are transported through the filaments and hollow central core and reach to the tip, and subunit is spontaneously aggregate, or ultimately flagella will made in bacteria. So flagellin is a subunit. They are transported through the filaments, hollow internal core, or ultimately flagella will form, or it will provide the movement of the microorganisms. Another thing I want to discuss, the various things which are available in the bacteria, like protoplasm. The cell wall enclosed to the protoplasm, which consist of the cytoplasm, ribosome, mesosome, granules, nucleoids, genome, and plasmid. So in case of the protoplasm, various things are there. Cell membrane, plasma membrane are there, you can say. So in the plasma membrane, cytoplasm are there, glucose and other number of the enzymes which are available in the protoplasm. Another is genome of the bacteria are there, basically genome which are present into the, it is a part of the protoplasm. Plasmid also is a part of to the protoplasm. Plasmid, it may be they are able to transfer their resistant, like antibiotic resistant and other drug resistant are there. They are able to transfer from the one cell to other cell with the help of the plasmid. It is a very important which are available in the bacteria, or with the help of this plasmid we can perform the genetic engineering or recombinant DNA technology. So it is a very important part of the bacteria. Ribosomes, it is a very small structures which are available in the protoplasm. They are able to synthesize basically 70s ribosome which are available. It is divided into the three part. So ribosome is which are available into the bacteria. Another various micro component which are available, they will help to the cell wall. And gas vesicles which are present, or ultimately it will help into the metabolism of the various products are there. Magnetosomes, the magnetosomes which are available into the bacteria. And another thing I want to discuss, the economic importance of the microorganisms. Beneficial, so I want to discuss some beneficial aspect of the bacteria, some harmful aspect of the bacteria. I want to discuss one by one. Nitrogen fixing bacteria, they convert atmospheric nitrogen into inorganic compound into the plant. Symbiotic bacteria, rhizobium is associated with the leguminous plants, and Frankia is associated with the certain dicotyledonous species. And free living bacteria like Enavina, Nostoc, Azotobacter, the number of the free living microorganisms, they are able to fix the nitrogen to the plant. Anaerobic nitrogen fixing bacteria which are also available in our atmosphere, like Clostridium pasturanium and Desulfovibrio vulgaris. So these are the two very important microorganisms. They don't require the oxygen, or ultimately they are able to fix the and nitrogen. And cyanobacteria, Nostoc, Anabina, Nostoc perform the this ammonification and nitrification is also, and some other beneficial aspect of the bacteria are there, like making of the curd, yogurt production, fermented food, sauerkraut fermentation, production of the single cell proteins, acetic acid production. These are the these things the bacteria they are able to form it. And uh, uh, another beneficial retting of the fibers and leather industry, biodegradable plastic, and production of the human insulin by recombinant DNA technology. In this case, E. coli, they are utilizing it, or ultimately they are able to produce the insulin. And cleaning of the oil spills, Pseudomonas batuta, they are able to clean the oil spill. And Bt cotton is transferred to the crop plant, resistant insect to. And harmful aspect, there are the various harmful bacteria are there, like denitrification, thiobacillus species, micrococcus reduce soil fertility by converting soil nitrate into atmospheric nitrogen. And blight disease of paddy, xanthomonas, the various harmful aspect of the bacteria are there. And bacteria cause the food spoilage, number of the bacteria they can cause the food spoilage. And biobar, bioterrorism. Dear learner, I want to summarize my second part. The bacteria in prokaryote microorganisms, they do not contain the true nucleus and unicellular. And bacteria are classified by shape and arrangement as the cocci, bacilli, vibrio, spirula, diplococcus, and cubicle packets. And bacteria affect human life both positively and negatively. So I hope you have learned number of the things, like related to the history of the microorganisms, and general account of the microorganisms, economic importance, various economic importance of the microorganism. You have seen the various harmful bacteria are there. Uh, if you have any question, if you want to study more, you can go through the BBYCT unit number 3. You can study it. Thank you.
Do you know that the world's population has reached 8 billion on 15th November 2022? And we are still counting. I don't know where do we go from here. The predictions are scary. Any idea?